Welcome to clinical research. The primary objective of this domain is to design and evaluate clinical trials that generate scientifically valid, reproducible, and compliant data. Clinical research is the scientific engine of drug development, turning laboratory discoveries into evidence-based medicines. Scientists and clinicians design studies to evaluate the safety, tolerability, dosing, and efficacy of investigational products. This work requires a deep understanding of disease biology, clinical trial methodology, and biostatistics to design trials that can withstand scientific and regulatory scrutiny. Clinical research teams work closely with ethics committees and regulatory agencies to ensure that studies are designed ethically and protect subject well-being. Throughout this lecture, we will explore the core methodologies, study designs, and clinical phases that define modern clinical research. The clinical protocol is the master design document for a clinical trial, defining the scientific objectives, methodology, and operational plan. The protocol outlines the primary and secondary endpoints that will measure safety and efficacy, and defines the inclusion and exclusion criteria that determine patient eligibility. It also specifies the schedule of assessments, detailing the timing of laboratory tests, physical exams, and drug administrations. A well-designed protocol ensures that all sites conduct the trial consistently, minimizing variability and bias. Developing the protocol requires collaboration between clinicians, biostatisticians, and clinical operations experts. Once finalized, the protocol is submitted to institutional review boards and regulatory authorities for approval. Any subsequent changes to the study design require formal protocol amendments and reapprovals. Phase 1 clinical trials represent the first in-human evaluation of an investigational product, focusing on safety, tolerability, and pharmacokinetics. These trials typically enroll a small group of healthy volunteers, ranging from 20 to 80 subjects, although oncology trials enroll patients with active disease. The primary objectives are to evaluate the safety profile, determine the maximum tolerated dose, and identify dose-limiting toxicities. Pharmacokinetic studies characterize the drug's absorption, distribution, metabolism, and excretion in the human body. Phase 1 trials utilize single ascending dose and multiple ascending dose designs to escalate the dose levels under close clinical supervision. The data generated in Phase 1 are critical for establishing safety boundaries and determining the initial dose levels for subsequent Phase 2 clinical trials. Phase 2 clinical trials assess the proof of concept and therapeutic efficacy of the investigational product in patient cohorts with the target disease. Typically enrolling between 100 and 300 subjects, Phase 2 studies are subcategorized into Phase IIA and Phase IIB. Phase IIA trials are exploratory studies designed to assess efficacy trends and early biological activity. Phase IIB studies are dose-ranging trials that aim to identify the optimal therapeutic dose for pivotal trials. Researchers monitor patient safety, evaluate dose-response relationships, and analyze secondary endpoints like biomarkers. The findings from Phase 2 trials confirm the therapeutic hypothesis, define the safety profile in the target population, and select the doses that will be evaluated in large Phase 3 studies. Phase 3 clinical trials are large-scale, multi-center, pivotal studies designed to confirm the therapeutic efficacy and safety of a drug in a broad patient population. Enrolling hundreds to thousands of patients across global clinical sites, Phase 3 trials generate the robust statistical evidence required by health authorities for marketing approval. These studies are comparative, evaluating the investigational drug against placebos or standard of care treatments to establish statistical superiority or non-inferiority. Clinical operations teams manage complex logistics, including global supply chain coordination, multi-site monitoring plans, and standardized data collection procedures. The cumulative safety and efficacy databases from Phase 3 trials form the cornerstone of new drug applications or biologics license applications submitted to the FDA, EMA, and other regulatory agencies. Phase 4 clinical trials, also referred to as post-marketing surveillance studies, occur after a drug has received regulatory approval and is available on the commercial market. These trials are conducted to monitor the long-term safety, real-world effectiveness, and optimal clinical use of the therapeutic agent in diverse, unselected patient populations. Phase 4 studies collect real-world evidence to detect rare or delayed adverse events that may not have been observed in smaller pre-approval trials. Additionally, these studies may explore new indications, assess health economics and outcomes research, and evaluate drug-drug interactions in patients with multiple comorbidities. The data gathered during Phase 4 surveillance provide continuous safety updates for the product label, support ongoing risk management plans, and help healthcare providers optimize treatment protocols based on long-term clinical data. Randomized controlled trials represent the gold standard of clinical study designs, utilizing randomization to distribute confounding variables evenly across treatment arms. Clinical operations teams coordinate randomization using interactive response technology to assign subjects to active or control groups. Randomization can be simple, blocked to maintain balance within site cohorts, or stratified to ensure key prognostic factors, such as age or disease severity, are balanced. Allocation concealment must be maintained until the assignment is finalized, preventing selection bias by ensuring investigators and subjects do not know the upcoming treatment group. By eliminating systematic bias and minimizing confounding factors, randomized controlled trials isolate the therapeutic effect of the investigational product, providing the highest level of internal validity and statistical reliability for clinical efficacy assessments. Blinding methodologies are critical study design elements implemented to prevent observational and reporting biases by withholding treatment assignment information from key participants. In single-blind trials, the subject is unaware of the treatment, while double-blind designs conceal the assignment from the subject, investigator, and clinical site staff. Triple-blind studies extend this concealment to the sponsor's clinical data coordinators and biostatisticians involved in data analysis. When active control or placebo comparators are used, matching placebos must replicate the appearance, taste, and packaging of the active drug to maintain the blind. The protocol must define strict guidelines for emergency unblinding, detailing the specific medical scenarios and authorization pathways required to break the blind via the RTSM system. This ensures that the blind is only broken when immediate clinical management of a serious adverse event requires knowledge of the treatment.